To inoculate, we use leaves of a tomato-susceptible variety that have been infected with our routine ToBRFV isolate. Those leaves carry a lot of virus particles and, when smashed with a liquid buffer, make the perfect infectious inoculum. By rubbing every single plant, the virus enters mechanically, ensuring an homogeneous infection across the whole greenhouse. A few days after the inoculation, susceptible checks are evaluated with immunostrips tests, which confirms virus development, which means a successful inoculation.
A few weeks after the inoculation, the first symptoms can be observed on the leaves. A series of evaluations of both leaves and fruits will take place, enabling the understanding of phenotypic disease progression in all the different pedigrees tested. In addition to the visual phenotypes, our team also collects plant tissue over time. That material is tested with quantitative PCR, enabling the measurement of virus accumulation during the whole cycle. The sample plate is lysed using a shaker. The prepared samples are then transferred on our liquid handling robot for automated RNA isolation. The total RNA present in the sample is isolated using magnetic bead capture technology and transferred to a qPCR reaction plate. This plate is loaded in a thermocycler for amplification. The signal generated is then analyzed. With our approach, on top of traditional agricultural performance, we aim to deliver hybrids that show no or minor ToBRFV symptoms, with reduced virus accumulation over time. The combination of early leaf symptoms and virus accumulation measurements on leaves have allowed us to select novel genetics that enable healthier crop development with zero or minor fruit symptoms. We have seen that the more we stack our novel genes, the less symptoms we observe on the fruits. As ToBRFV is a quarantine virus, we follow the local government's regulation in order to do our research. The waste removal is a critical step, since we don't want the virus to spread out of the contained area. That is why all waste, substrate, plants, labels, etc. must be incinerated by a certified third party. The material is placed in sealed plastic containers that can be safely transported to the incinerator without leaking. After discarding, the whole facility is disinfected with viricide and rinsed with clean water. Then we are ready for a new cycle of tests.